Hi, I'm Stacy Lindborg, Executive Vice President and Head of Global Clinical Research for BrainStorm Cell Therapeutics. BrainStorm is a leader in developing innovative autologous cell therapies for highly debilitating neurodegenerative diseases. The origins of BrainStorm's cell therapeutic platform and the most advanced technology in our portfolio today is an autologous cellular therapy named Neuron. Neuron is an autologous product, which means that we use each patient's own cells to produce their product. This patient is convenient, it's consistent, and it's reliable. By convenient, as part of the manufacturing process, we cryopreserve Neuron, which allows us to create an off-the-shelf product for each patient. One harvest of bone marrow creates several years of therapy. And from the point of harvesting each patient's bone marrow, which is a relatively simple outpatient procedure, the first treatment is available in 19 days, with subsequent treatments being available in only 7 days. Attributes that contribute to the product being consistent and reliable include that there are no animal proteins, antibiotics, genetic modifications, or viral vectors used in the production of Neuron. By using culture rescue, we create a product with very high cell viability and consistent performance characteristics. And of course, Neuron is manufactured with cell potency release criteria, which is important to the reliability of the product. Across the last 10 years, we have established the relevance of Neuron across neurodegenerative diseases. And we're expanding our R&D portfolio to include both allergenic as well as autologous products by developing an additional technology harnessing exosomes induced from Neuron. Each product is delivered in a manner that ensures high biodistribution and efficacy. Neuron, for example, is delivered intrathecally, which enables distribution through cerebrospinal fluid, or CSF, providing direct access to the brain. And our exosome product that is produced from Neuron will be developed for intratracheal delivery. We've taken the best of cell therapy and applied this to neurodegenerative disease. From a platform perspective, BrainStorm has been developing Neuron technology for 14 years with numerous animal studies and animal models of various neurodegenerative diseases that you can see on this slide. Across these studies, we've observed consistent biology with in vitro and in vivo data, and more recently, through CSF biomarker data and clinical trials. We've completed clinical trials in 2 different neurodegenerative diseases, ALS and progressive MS, and we're establishing the next steps for both of these diseases. Additionally, we will soon be starting a trial in Alzheimer's disease. We continue to broaden our portfolio, and as referenced earlier, we are progressing preclinical research in our exosome technology, which is derived from Neuron. The additional trials that we anticipate starting will continue to accumulate evidence around our technology and broaden the understanding of our mechanism of action. Turning to our most progressed clinical development program in the relentless disease of ALS, we have been developing Neuron technology in the clinic with ALS patients for 11 years. We conducted a comprehensive and carefully thought out program that comprises an open label phase 2A study, a randomized phase 2 study, along with a recently completed phase 3 study. Both of the earlier trials have been published in peer-reviewed journals and are available on our corporate website if you'd like access. A phase 3 trial in ALS patients was completed at the end of 2020. We failed to reach statistical significance on the primary endpoint, yet pre-specified analyses suggest that there are clinical benefits for a subset of patients. An important part of establishing the next steps for Neuron and ALS will be to publish a comprehensive review of the findings from phase 3. In fact, a manuscript focusing on the clinical data is currently under review, and a manuscript focused on the rich biomarker data is set to follow shortly. We remain committed to pursuing the fastest and best path forward for Neuron. Across our clinical portfolio, we understand the importance of establishing and working with world-class manufacturing partners, leveraging the best treatment centers with world-renowned investigators, and incorporating the voice of the patient in all that we do. For ALS, this meant working with City of Hope and Dana-Farber to produce Neuron, which included more than 800 shipments for the phase 3 trial alone. This also meant working with 6 treatment centers across the US and their world-renowned ALS clinicians and study investigators. And finally, this meant partnering with preeminent patient advocacy organizations and organizations that are committed to accelerating stem cell therapies to patients with unmet medical need. We had funding for this trial to further biomarker research in ALS from the California Institute of Regenerative Medicine, from the ALS Association, and IMALS. And we continue to partner with these funding agencies as we seek to deliver an innovative therapy for ALS. For all clinical diseases we study, we'll apply the same rigor in establishing the best partners to help BrainStorm innovate and deliver therapies for patients in need. Let me say a little bit about our new technology leveraging exosomes. So exosomes are nano-sized secreted vesicles that are released by different cell types. BrainStorm's new technology leverages exosomes secreted from mesenchymal stem cells, which we call MSC-NTF exosomes. Exosomes contain cargo, which includes microRNA, messenger RNA, lipids, and proteins that really come from the cells of origin. And they participate in cell-to-cell communication to deliver their content, ultimately providing regulatory functions for many different cell processes, including inflammation, angiogenesis, and regeneration and differentiation. By encapsulating molecules within their membranes, exosomes can protect proteins and micro-messenger RNA against degradation. Exosomes have practical advantages compared to cell therapies and are a great addition to our portfolio as a result. For example, the ability to effectively cross biological barriers, such as the blood-brain barrier, and penetrate into deep tissues. They have high product stability and easy control of product formulation, which may enhance the expected therapeutic benefits. They have low immunogenicity, and they possess ideal innate structural and carrier properties, enabling transportation of low molecular weight therapeutics, nucleic acids, and proteins. We've done an extensive amount of research, which I can't adequately cover today, but I do want to note that we have a recently published paper which examined 2 different acute lung injury models in mice. In this paper, which is available on our corporate website, we demonstrated beneficial effects of Neuron exosomes and their superiority to exosomes from mesenchymal stem cells. These were across multiple parameters, including increasing blood oxygen saturation and reduction of lung pathology. For more information, I'd refer you to our paper. But let me finish with my comments on exosomes, that this really is a promising area of therapeutic research for many diseases, including lung and various eye pathologies. And it is an area of investment for BrainStorm. The bedrock of BrainStorm lies in our state-of-the-art cell therapy manufacturing capabilities. So what differentiates us, and what are our recent accomplishments? Well, Neuron originates from Tel Aviv University, and we continue to build on strengths from our R&D and manufacturing center in Israel, including the leadership of Dr. Yosef Levy, our head of manufacturing, who has been with our technology since his Ph.D. research. At BrainStorm, we have established a reliable supply chain across our clinical portfolio, enabling production of around 400 products for trials both in Israel and in the US. On top of manufacturing partnerships which were established for clinical trials with Dana-Farber and City of Hope, we have 2 GMP certified clean rooms in Israel, which expand our geographic range beyond the US. We have established Catalent as a commercial manufacturing partner and a partner for future clinical trials. And additionally, we have ongoing CMC development with bioreactors to enable automation and additional CMC efficiencies. When we look at our senior leadership team, you can see that we've complemented the deep experience in R&D and manufacturing from Israel with experience in the US covering commercial, pricing and access, patient and government affairs, and R&D. We will continue to be nimble as a company, leveraging consultants and contract relationships, in addition to growing our employee base, adding additional expertise needed to launch us into our full potential as a company, which at the heart is meeting incredibly urgent patient needs and delivering commercial growth to propel BrainStorm forward with future R&D. We have some exciting science coming out in the near future at important international meetings, really across the next couple of months. At the 37th Congress of ECRMS, Dr. Jeff Cohen from Cleveland Clinic and a principal investigator in our progressive MS Neuron clinical trial will be giving an overview of the results out of this trial. Dr. Robert Brown from University of Massachusetts and PI in our phase 3 ALS Neuron clinical trial will be covering biomarker results based on this trial at the 32nd International Symposium on ALS and MND. And then lastly, we will be releasing some new and exciting results from our exosome technology at the New York Stem Cell Foundation Conference. So thank you for showing interest in BrainStorm, and we look forward to coming back to you and giving you future updates.